strawberry breeding lead for Driscoll's. So we have a real rare opportunity to hear from one of the world's experts in strawberry breeding. Phil, I had a chance to talk to him about his past, and I know he's going to, not that past, but the educational past, and what his educational background is. It's always interesting to hear how people got to where they are. And Phil started out with a BS in biology. He's from upstate New York, and Alfred University in upstate New York, where he got his bachelor's degree, and then went to University of Arkansas to do a master's degree in breeding on blackberries, and then a PhD at University of Florida working on strawberries. And then from there was actually recruited before he graduated to work at Driscoll's, and he's been at Driscoll's for 12 years as a strawberry breeder. So there's good opportunities, job opportunities in breeding, and this is a really exciting area no matter what crop you're working on. I think the ability to manipulate the genetics of the plant and to get it to do things that we all want them to do. And Phil's going to talk to us about what those objectives are. So with that, Phil.
Oh, thanks. So I'm going to repeat a little bit, although maybe I'll just skip through a lot of it because Gerald just covered a lot of my first part here, but I wanted to talk a little bit about my background, and I think you're all going to be in the position of figuring out what you want to do with your careers, so maybe you can it's either a cautionary tale or maybe something to aspire to, depending how you look at it. I'm going to talk a little bit about kind of the history of strawberry improvement, then a little bit more about how modern breeding programs work, and then specifically about the Driscoll's breeding programs, and then some of the technologies we're using. This was, a lecture on strawberry breeding is, it's right up my alley, but it's sufficiently broad as to, first it seems like, oh, I could do all kinds of things, and then you realize it's broad enough that I don't know how to cover all of it. So I haven't gone super in depth on all of these things. I'm hopeful we have some time to talk at the end. And if you got questions, I'm happy to fill in details or clarify things. Just let me know. So the question is, how the heck do you wind up breeding strawberries for a living? And I kind of ask myself the same thing once in a while. I don't know, are any of you familiar with plant breeding? Have any coursework or work with it? So I was in the same position as most of you up to about a week before graduation. I went to Alfred University, which is a little liberal arts school in upstate New York. That's me in my college years. It's actually the statue of King Alfred, and I didn't put that pumpkin there, but I put another pumpkin there an earlier year. I wound up in biology because I liked science and I knew I didn't like chemistry, so I figured biology is not chemistry, so that works. And I like plant biology a lot. But I was about to graduate. I had no job prospects, really, and I ran into a relative who had a job in entomology at Cornell, and I applied for that, and I didn't get that job, but my application was on file there, and the grape breeding program contacted me, and I figured, I don't know what grape breeding is, but I do like being employed, and so I took that job, and I wound up in Cornell for 5 years working in the grape breeding program with Bruce Reich. He was my boss. It's the guy holding the grapes there. And so, I think I took a little bit different route into breeding because I learned it on the job rather than in grad school. And I think there's pros and cons. There's definitely a lot of things I missed because of this. But I also feel like when I got to grad school, I kind of figured out a lot of the basic concepts, and I had a little bit of an unfair advantage over some of the other students in that regard. From there, I went on to Arkansas, and I got a master's working in blackberries with John Clark. And I worked with disease resistance breeding for 2 diseases in blackberries. And as I was telling Omar earlier, a lot of the coursework I took at that point was kind of making up for the horticulture coursework I didn't do as an undergrad. But I got a lot of plant breeding as well.
I'm curious what the 2 diseases were.
Erwinia amylavera, so fire blight, which is well known in apples and pears and virtually unknown in blackberries. I'm actually author of half the publications on blackberry fire blight, which you could do in the course of a master's degree, which gives you the sense of how much it was. And then Botryosphaeria dithydia. So I was telling people earlier, I was hired and they said, we had this terrible mystery disease in blackberries last year, and your project will be to figure out what it is and look for resistance to it. And I started, and the mystery disease didn't come back. So I went around and I tried to culture every last little bit of sick tissue I could get, and those are the 2 diseases I found, so that's what I kind of defaulted into. So I wound up working on a disease that nobody does research in.
Never came back?
Not as far as I know.
Okay, interesting.
So in Florida, I went to Florida then for my PhD, and I worked with both the strawberry breeder and Kevin Folta, who's in this picture. He's a molecular biologist, and I focused on the genetics controlling photoperiodic flowering in strawberry. And one of the things we looked at was different light quality. So he's got different chambers there of different colors with LEDs. And then from there, Driscoll's recruited me basically right out of grad school. In fact, I finished grad school early to go to Driscoll's because it was kind of my dream job, and I was determined that despite the fact I wasn't planning on graduating for a year, I was going to make it happen. So I started out in charge of Central California, Central Florida, and Central Mexico, which we jokingly called the Central District strawberry breeder. And then over time, I took over most of the strawberry breeding work. So just a little background on the strawberry itself. The cultivated strawberry that we grow is actually a hybrid of 2 species, both of which were brought from the Americas to Europe. One of them is Fragaria virginiana, which probably, it's all over eastern North America, but the specific one involved probably came from Canada. And the other is Fragaria chiloensis, which grows all up and down the west coast of North and South America, although the North and South American subspecies are pretty different. And in South America, it's actually cultivated some, just selections from the wild, and this is one of them. They're round, firm, they don't color very well. The natives there had been cultivating the white form for a long time. And so actually a French spy sent to the Spanish colony in Chile saw these strawberries, which were much larger than anything growing in Europe, and thought they were pretty cool, decided to bring some back. He brought 5 plants back, gave 1 to the captain on the ship in exchange for the extra water he gave him to water the plants, and the rest went to a botanic garden in France where they promptly never set fruit. And at the time, nobody knew why, because nobody had figured out that there actually are different sexes in some plants. And so he had happened to bring all female plants. Because he chose ones he liked the fruit on. So it was sort of this mystery, and it was kind of a botanical curiosity. It floated around botanic gardens until eventually somebody planted it next to the Fragaria virginiana that had been brought from Canada. And it formed these hybrids, which were just kind of an accident. Somebody happened to plant the seeds out. And the very first Fragaria ananassa, which is the cultivated strawberry we grow now, looked a lot like this. It was a big lumpy thing, and it was probably pink, because it was a hybrid between a red virginiana and a white chiloensis. And so we really could have wound up with white strawberries, except that the wild strawberries in Europe were red, and Europeans didn't like pink strawberries. They wanted them to look red, so they bred them back to being red. So this is a very easy to understand graph. This came from Steve Knapp's group at UC Davis. And I don't expect you to, figure it out, but I just wanted to sort of emphasize, if you look up here, there's a really narrow base to the genetics of strawberries. Almost everything comes from just a handful of things. And all that comes down, and it's like, yes, we have a lot of different varieties now, but they all trace back to just a real narrow group of things up there. So if you look at where the actual ancestry of these things comes from, if you look by different regions, 3, 5, 4, 7 founders make up more than 50% of the genes in each of these regions. And if you look at the total number of ancestors, this is the absolute maximum. Some of these are actually related to each other, so they'd actually be less. But somehow, despite that narrow genetic base, we keep making progress. So this is from my program. It's an old graph, but you can see the graph is expressed as percent of a standard, which is the variety San Juan. And you can see we've gone from an average of 80% up to an average of 120% in 10 years, which that's a pretty good gain, if I do say so myself. A lot of the agronomic crops would be really happy with that kind of yield gain in that time. So despite this really narrow genetic base, we seem to still be able to select better and better things. And part of that is the way the genome of strawberry is set up. So most things, you, me, the majority of plants and animals are diploid. So we've got, strawberry has 7 chromosomes, or 7 pairs of chromosomes. The diploid wild strawberry, and you and I have pairs of chromosomes, but the cultivated strawberry actually has 8 copies of every chromosome. And so one implication of this is that despite having this relatively limited number of founders, we have a lot of different copies of every gene that can vary. So there's a lot of sort of hidden variation in there. I do think at some point we do have to be concerned about lack of variation in our programs, but the Octoploid genome hides a lot of diversity. It makes a lot of the work we do on molecular tools much more complicated, which I'll talk about a little bit later. So I figured I would just talk a little briefly about our objectives, and I think really almost every strawberry breeding program in the world is going to have a pretty similar list. So at the top of the list, like it or not, is yield. That's just an economic reality. Timing is really important. Depending on where you're growing, the production in a certain window is worth a whole lot more than in another window. So, for example, in Watsonville, where I run my program, late yield is really where people make their money. So you want something that finishes the season strong and just keeps pumping out fruit late into the season. And that yield's worth a lot more than yield in like the first week of July when prices are really low. In Oxnard or Florida, it's early yield that's valuable. So it's not just about sheer amount of yield. Also important is flavor, and at Driscoll's we see ourselves as a premium brand, and sometimes we do better against that objective than others, but in general we have put a real emphasis on flavor. Flavor is a really hard objective to work with. I'll talk a little bit more about it later, but yield, you can take every fruit that comes off it and weigh it. You get a number. It's a real number. It's real data. You can run stats on it. Flavor is a series of subjective opinions. So, what you think about something and what I think about something are equally valid, but they're not the same necessarily. We have different preferences. We have different cultural backgrounds. We have potentially different genetics that influence how we taste things. And so, a lot of what we deal with flavor and in the breeding program winds up just being averages. You're trying to pull a lot of people together and just sort of, try to find things that make most people happy. Shelf life is another one. Obviously, most of the fruit grown on the West Coast goes to the East Coast, which 50 years ago was really challenging. Now it's kind of routine, and we spent a lot of time looking at that. Disease resistance can be a big factor. Sometimes really susceptible things can succeed, but it can kill a variety too. Plant habit has kind of come out of nowhere. We didn't worry about it very much when I started, but with labor getting scarcer and more expensive in California, a plant that people want to pick is really valuable. So you want fruit that's well exposed, and the other aspect of plant habit is air circulation, which is has implications for shelf life and disease as well. But probably more than anything, although maybe it's really a combination of all these things, is adaptation. And so we have a lot of strawberry breeding programs, and I get asked a lot, like, why can't you just use the varieties from Watsonville in Florida or Santa Maria or Oxnard? And it's because strawberries are really narrowly adapted. So I just put this up here as an example. So we've got the blue lines are 3 varieties from the Watsonville breeding program. The yellow are 3 from the Florida breeding program, and the red are 3 from the Oxnard breeding program. And then you can see how they've performed. This is yield in Watsonville, Florida, and Oxnard. And you can see the same varieties that do really well in Watsonville do just terrible in Florida. And on top of that, with the timing thing I mentioned before, they're super late. You wouldn't make any money on these. And the same thing is really true in all these places. The Florida ones are probably the most broadly adapted, but they really don't like our soils in California, particularly the real heavy soils, because they're bred on like pure sand, and they're used to just sort of being spoon-fed irrigation and nutrients, and they just get super stressed out on our heavy soils that we have. So the other factor involved, well, one of the factors involved is flower initiation. That's one of the big determining factors of what works where. And flower initiation in strawberries is really controlled by 2 things, day length and temperature. And you can see this, so this is a variety called Abundance, and you can see at 24 degrees Celsius, it barely sets any flowers, even in really short days. As the temperature goes down, it will set flowers in slightly longer days, and if you get to a real low temperature, it'll actually set flowers in 24-hour daylight. And that's one of the things that makes California, and especially Watsonville and Santa Maria, really good places to grow strawberries, because we have cool summers, and so the cool temperatures allow you to keep producing flowers through the long days of summer. Some of the other things that impact adaptation, I mentioned soil type. Chilling is an interesting one. There's a pretty big difference in the amount of chill in Watsonville versus Oxnard. And chilling affects kind of the vegetative balance of the plant. So if you get more chill, you're going to get a bigger, more vegetative plant. If you get less chill, you're going to get a smaller plant. And so you're trying to find a balance that gives you the right balance between leaves and fruit. And if you overshoot, you wind up with a big plant no one wants to pick. It doesn't make much fruit, and you get lots of runners. If you undershoot, you get a little weak plant that can't actually ripen the fruit that it makes. So because of this, there are a lot of strawberry breeding programs. I have an Excel file on my computer that lists a bunch of them. I don't know that this is an exhaustive list, but I put a dot on the map for every breeding program I had. And you can see they're all over the world. And Driscoll's is not quite as widespread, but we've got a lot too. So how do we actually do the breeding? So the breeding process starts with a cross. And I actually was going to do a YouTube video, and I realized I have no internet, but I'll talk through it. We
can set it up if you need to.
Well, we can set it up at the end while we're talking. So, a cross is, that's what breeding is really fundamentally about, is making hybrids. So you start with the flower. I don't know if you've all had plant anatomy kind of things, but you may or may not be familiar with these terms. But basically, every plant in cultivated strawberries is both male and female. So you've got, these are the female parts with the style and the stigma, and they connect to these ovaries, and the ovaries will develop into the achenes, what we'd call seeds, on the outside of the strawberry, and actually it's this receptacle tissue that develops into the fruit that we eat. And then over here is the anther, which is part of the stamen, which is the male part of the plant. And so when we make a cross, it's pretty simple. At least it's simple in concept. It takes a little manual dexterity to do it. But basically, we remove the male part. So we peel these things off, and the guys on my crew who do it can do it lightning fast, and I do a bad job, and it takes me forever. And then we take pollen from the other parent, which is being shed by these male parts, the anthers. And you can see, it's kind of hard to see, but there's pollen being shed off each of these things. And it's just as simple as taking the pollen from the male parent and putting it on these stigmas of the female, and then which fertilizes those ovaries, and that seed will be hybrids. So the crossing itself is pretty simple. Usually it works, no matter what you choose as parents. But choosing what parents you choose obviously is critical. And in general, we're trying to choose parents with complementary characteristics. So, if you've got something that is high yielding and disease resistant, but tastes terrible, you might cross it to something that tastes good. You're trying to stack beneficial traits, which sounds pretty simple, but we had a long list of traits there that we're selecting for. There's really, you could probably come up with 20, 30 more. So there's a lot of things being evaluated. So it's really kind of a balancing act, and every cross and every selection you make is a compromise. We do learn from experience, so sometimes things that have good traits turn out to be terrible parents. So, I like to think I do learn from my mistakes once in a while. You can also predict to some extent which things are going to be good parents by using quantitative genetic methods to calculate breeding values and heritabilities and general combining ability, that kind of thing. And we also have molecular markers, which I'll talk about in a little while later. So breeding programs vary a great deal in their approach to cross number and scale. We do about 700 crosses a year with an average of about 150 seedlings. Individual crosses might be from like 45 to 400. I've actually been as high as 1,000, though that was a mistake. But there are some programs, it's really different from program to program. So like the Queensland, Australia program, I was talking to the breeder there, they average like 30 seedlings across. I also talked to a breeder in China who did 5,000 seedlings from a single cross, which seems insane to me because that's like an entire breeding program. But if you know what you want, you can increase your odds of getting it, I guess. So once we've made that cross, at first you can't really tell whether you succeeded, but if you have, it'll develop into just a regular strawberry. That goes in a blender, and we blend that, add a little water, add a little pectinase, which is an enzyme that breaks down the pectin and allows it to kind of move, because otherwise it's pretty gloppy. The good seeds will sink, the bad seeds and all the detritus float, so you sort of skim that off, and then we dry down the seeds and we sow them in these flats. They spend a little time in the refrigerator gathering some chill, which helps them germinate. And so we sow them out in these flats, they start to germinate. And then we transplant them into these cell trays, and they're 48 cell trays, which actually kind of matters because when we do the molecular markers, those are 96 well plates. So two 48 cell trays line up really nicely with that, so you can it's easy to figure out where a well in the plate lines up with a cell tray. And they'll just transplant seedlings from that germination tray into these cell trays until we hit the number that the breeder has asked for. So the process at Driscoll's it's basically a 5-stage process plus a commercial trial period. And really, it's a process of narrowing down, because the real challenge in breeding is a numbers game. So the higher a number you start with, the better your chances of having what you're looking for in there. But the higher the number you start with, the more it costs, the more time it takes, the more resources it requires to evaluate it. So the whole thing is a sort of a step-by-step process of eliminating more and more selections at each generation so that you can spend more time evaluating the ones that you think you care about. So we'll start with 25,000 seedlings. This is the Watsonville program. So we start with 25,000 seedlings, and we select down to basically, on average, about one at the end of the process.
That first stage, when you go, maybe you're going to talk about this, but taking it from 25,000 to 250, what's that based on?
So that's actually the next slide. At the seedling stage, it's probably the most subjective of all the stages. My team and I, so I've got 3 people who work with me, and we'll soon have someone else probably helping out. We walk these every week, and if we see something we like, we mark it with, we have a barcode sticker that we put on there. And actually, got that there. I'm kind of going out of order here, but we can scan that barcode, and it brings up a record for that variety on our phones that we can rate things. And every week when we walk past, we'll scan that barcode again and take another week of data. We're looking for all those things really that I listed on those objectives, but they're pretty subjective measurements. We're not weighing any fruit. We're not measuring shelf life. We're just saying it looks like a lot of fruit. It's got big fruit. And health is part of it. And I made a note here. We plant our seedlings in non-fumigated ground. And at this point, most populations are pretty tolerant of the soil diseases that we have, but every once in a while we come across a population that has issues, and I saw this one the other day and thought it makes a good example of what's going on here. So the world is, at least the world in California, is not going to have more fumigants in the future. It's going to have less. And so I want to push the program in a direction which takes it towards varieties that need less fumigants. So with the seedlings, we select up to 2% during the course of the season. And then about midsummer, I start whittling it down. Because one of the challenging things about strawberries is it's not a single harvest like a lot of crops. So you need something that can maintain quality and health and productivity over a season that's many months long. So there's a lot of stuff that looks good at the beginning, and by the time you get to the end, it's not producing, or the fruit size has fallen way down. So it's a lot of the things that look good at one point won't look good the whole time. So we want to make sure we select enough to have something to work with. And the other thing that's happening is we're sending this stuff, we're letting the nursery know what we're selecting at this point, so the nursery can start propagating it. So the nursery will plant out all that stuff that we selected, the 2%, but by the end of the year, we're going to throw out half of it. So stage 1 is the next stage. This is really where we start taking actual hard data. So these are 10 plant plots. This is also in non-fumigated ground. We bring in material from the other programs here. So like in Watsonville, we'll test the stage 1s from Florida and from Santa Maria as well. And in a lot of ways, we evaluate this just like the seedlings. We use almost the same form on our phones. The difference is that we actually take yield data. We take actual shelf life evaluations. And usually at this point, we start getting some molecular data as well. So from this level, we choose about 25 individuals to go on. And I find this one of the hardest cuts to make. With the seedlings, most of them are garbage. You can walk down the row and only a couple will really catch your eye. In the stage ones, most things look pretty good. And there'll be some that are pretty easy to get rid of, but I usually find I get it down to maybe 60, and those last cuts are pretty hard because everything looks pretty great. I'm wrong about that sometimes, but they do look great at that stage. So stage 2 and 3 are almost the same, really. These move to larger plots and more locations. So they're 40 plant plots, and we'll plant 8 to 12 of them per site, and that'll have a range of plant dates that they get planted on, because we're starting to think about finding that optimal plant date for growers. At stage 2, we plant them on 1 additional site, and then stage 3 is pretty much the same thing, but we'll plant it at 4 additional sites. Because the plots are much larger, the yield data at this point is much more accurate. This is where, at stage 1, that yield data, you can eliminate something as having terrible yield at that point, but it's hard to say if it's great yield or just okay yield, because on 10 plants, you don't have the resolution. If one of them got planted funny, maybe you're down 5% of your yield just from that. So in addition to the data that we took at stage 1, at this point we start flavor testing, and we do a lot of it. So every single variety, all 25 of those, plus our commercial varieties, plus the things in the higher stages, all go into weekly flavor tests. In Watsonville, that's done around the Driscoll's offices, because we've got a lot of people who work in Watsonville. And by the time something is actually released, it may be tasted 1,000 times. So it's pretty accurate. Doesn't mean there aren't big differences in opinion. I had a little friendly argument with our CEO about a variety that I dislike that he likes, that he probably saved me from dropping. So flavor's tough to evaluate, and I wish we could do more with it at earlier stages, although we are tasting them ourselves then, but this is really the point where we start getting real data. Let's see. So stage 4 is a lot like stage 3. The difference here is that we start adding cultural trials. And we do this, we used to do these ourselves, now we cooperate with an applied research group. These really, it depends on the variety. We'll look at the variety, kind of figure out what its weaknesses are or where it could use some optimization. So these might be spacing trials, they might be fertility trials, At this point, we also start testing them. Actually, at the previous stage, we start testing them in tabletops, which I don't know if you're familiar with, but they're well, you saw them on your field trip. So there's a lot of thought in Driscoll's that tabletops are kind of the future. So every one of our breeding programs now has at least a small test plot in tabletops in addition to the soil. At this point, we test everything at this stage for resistance to all the major pathogens. A lot of them we already have an idea because we have molecular markers. But even if we've got a marker for it, I prefer to have an actual result, because this is going to go out to growers at the next stage, and I don't want to tell growers something is resistant to a disease without actually testing it with that disease. And we also start working closely with the business side of the company with this. I guess it's all business, but the commercial fruit producing side of things. Because this is going to be a commercial variety next year, and I don't want anybody to be surprised by any aspect of it. So we start looking at whether this can go for export, because that's a big part of our business, and they've got different requirements. We start sending fruit to the QA inspectors, and I start talking about it a lot. It's not that I keep it secret beforehand, but talking about it a lot to growers and other employees, just getting people ready to think about it and be ready to grow it and deal with the fruit next year. So the commercial trial, we do a big commercial trial. I think it's one of the things that's really different for the way Driscoll's breeding programs handle this. Our commercial trial is 2% of the acres in a district. So, if we've got about 5,000 acres in Watsonville and Salinas, this would be a 100-acre trial. And that gets spread out across a number of growers. And it's really about trying to see as much as we can, as fast as we can, and trying to get growers comfortable with a variety quickly so that we can move on it. So we turn over varieties really fast. The average lifespan of a variety is 3.3 years, and there's a new variety almost every year. So, it doesn't really, we're not using our resources wisely if we can't move fast. So this way we can see it on a bunch of different ranches and a bunch of different microclimates and soil types and management philosophies. It gives growers a chance to get comfortable with it. And it gets us comfortable enough to go to big acres the next year if we want. And then the final stage is a commercial cultivar. And if it meets all our requirements, hopefully this is what happens to it. Some varieties are kind of bit players. They hang around and they're 5–10% for a few years and then they go away. Other varieties will last. I'm hoping I don't have any last this long in the future, because it's a sign that maybe I'm not advancing other stuff fast enough. But we've had varieties last 20 years. I think the longest running one since I've been there was San Juan, which was released in 1998 and finished in 2001. So we wind up naming a variety roughly every other year, a little bit more than every other year. So the other thing, and I don't really have time to go into detail, but I want to hit on it because I think it's like so often forgotten in breeding programs, is the breeding nursery. The breeding nursery is like the hidden half of the breeding program. So the reason we can move as fast as we can is that running in parallel to the breeding program is the nursery. And the nursery is making sure we have enough plants at every stage to do what we want, and that when we get to the commercial release point, there are millions of plants there ready to go. And we can go 5 years from a single plant to acres because we're willing to make the investments in this. And I think a lot of breeding programs don't fully appreciate how important that is. So there's a few other tools we use, and I've mentioned some of them before. I don't need to spend a lot of time on them, but test plot data is a big part of what we do. Looking at things in test plots are a big part of what we do. But there's some other tools that help us get better data. One of them is disease and pest assays. The trials here have been a part of that. I don't know if they've seen your trials here, but the Macrophamina and Verticillium and Calatotricum trials, we've got varieties in that. They're evaluating. We have our own pathology group that also evaluates things. Sometimes you can just look at the test plot and it just gets the variety, and you're either lucky or unlucky, depending how you look at it. But the absence of disease in your test plot doesn't necessarily mean that it's resistant. So sometimes you have to do controlled testing. Some assays are conducted in a greenhouse. Some assays, and this is actually here at Cal Poly, are conducted in a field, either a field that's known to be infested with a pathogen, or a field where you've inoculated the plants ahead of time. So, and then the other way we do pathology, and it's actually probably the way the majority of our pathology information comes, is through molecular markers. Right now, through either inoculation tests or markers or both, we're assaying for resistance to all of these things. I actually left Xanthomonas off of there, but not all of these are a problem everywhere, but I'd rather know and not need it than need it and not know. So another thing that we look at that really has been an area where we've grown a lot in Driscoll's is the chemistry and sensory side of things. So our flavor tests will tell us how much somebody likes something, but it won't necessarily tell us why. It may be that you like, the fruity aroma, or you like the sweetness, or maybe you don't like it, and you don't like it because it's mealy, or the color's blotchy, or it smells like dirty diapers. We actually have strawberries like that. And so these sensory studies allow us to go beyond just a simple flavor rating and break it down into components and tell us what's important and what's not, and where we can focus our efforts. So the sensory side of things, this is I don't know if you've ever looked at a flavor wheel or aroma wheel. This is from wine. We're actually working on them for strawberries right now. I was hoping I'd have one, but Henry, our sensory specialist, got a little nervous about, he said it's not ready for prime time. So, but sensory evaluations can look at, we talk a lot about aroma, but really there's appearance, there's texture, sweetness is a huge one, obviously, for strawberries, sourness. So once we break it down, we can know what to focus on. And then we can take it to the chemistry side of things. So once we know that, it's dirty diaper aroma that we want to eliminate, or, increase sweetness, we need to know, where does that come from? We need to know what actual compounds are involved. And so we go to the chemistry group, and from that, we generate information that we can correlate with that sensory information. So this is GCMS, gas chromatography, mass spectrometry. I can't say it, spectrometry. Yeah, you know what I'm saying. GCMS. And this really is looking at the volatiles in a strawberry. And you can see there's 3 different varieties. And for example, this peak is present in this variety, but not in these. And then you have other peaks like this one, which is present here, but taller, and present here, but shorter. So that's a quantity difference. So we take that, and we can go to our molecular group. We can also just learn from that. But ultimately, we try to take everything to the molecular group. This is what we call GWAS. This is genome-wide association study. And basically, we sequence all the varieties, we take their genomes, and we just look at every single variant, every base that varies in that genome, and we say, is there a statistically significant association between this base and the trait? And you can see, in this case, this is for the ever-bearing trait, actually. It's pretty clear that this area is much more strongly associated with that trait than the rest of the genome. And so, basically we're looking to find characteristic pieces of DNA sequence that associate consistently with certain traits. And so we have these for disease resistance, for flavor components, production patterns, flowering habit, all kinds of things. And we're adding to this all the time. We can, like I said, for the disease, we can use these as a proxy for where generating phenotypic information is expensive or difficult or time-consuming. So you can see this is a resistance marker. If the G is the resistant variant, these have the AG genome marker scores at that locus. And so they have the G, they're resistant. These have 2 A's, they're not resistant. It also can help us plan crosses. So if you wanted to plan a cross for that trait where G is resistant, and these were your varieties, which would you choose? Anybody want to make a guess? A pair of parents you might want to combine? Yeah. So the nice thing about D is that you're going to get a G no matter what. So all of your seedlings from that cross are going to be resistant. So if B, for example, has some other trait that you're really interested in, if you cross it to G, you're not going to have to worry about the resistance. If you cross it to A, for example, half those things are going to get the A allele, half are going to get G, so only half will be resistant. So I look at these for a lot of different traits when I'm planning my crosses. It's kind of one of these things where you just wind up making a lot of compromises. And then we also use these markers to screen seedlings. So we'll do 25,000 seedlings in Watsonville that actually make it to the field, but we'll actually grow more like 35,000. And I told you about the 48 cell traits lining up with the plates. This is how our group sorts it. So we'll run the markers, and we'll get this printout, and the green ones are plants that they save, and the red ones are plants that they throw out. And they've got the rows labeled here so they can see them, and they will just go through here and throw out things that we know we wouldn't have selected anyway because they don't have the traits that we want. This lets us use our resources more efficiently because, it costs a lot more money to grow a seedling in the field, farm it, look at it, on a weekly basis than it does to just grow it to a couple leaves and then throw it out because we know it doesn't have what we're looking for. So in the end, if we did everything right, the final result will be a variety that makes the consumer happy that they ate that berry and that they want to buy more. I saw a quote from Gene Galetta, who was the breeder of the USDA in Maryland, and he said his goal was to make a berry that if people ate it, they would be happy they were alive on that day. So I like that. And the other aspect, which is why I have a job, is to make Driscoll's growers profitable and Driscoll's profitable. So hopefully everybody wins, and that's it. Thanks.
Questions for Dr. Stewart?
Is it your initial crops? Because you mentioned it's 3 years for a new variety. Is it your initial crops, or do you keep doing generations?
So strawberries are a little different than some crops. Like some crops, you would take it through multiple generations to try to fix the genes, and if you're doing seed propagated things. In strawberries, because they're clonally propagated, they're highly heterozygous, and so you make that cross and you can propagate that initial seedling indefinitely. So it's really nice in a sense in that if you get something you like, that's all you have to do. It's less convenient in the sense that because everything is really heterozygous, every cross you make, everything segregates, and so you can't fix a trait neatly like you could in a seed propagated crop.
So every year it's another 25,000 seedlings.
Yeah.
25,000 crosses.
Yeah, so this process is running every year. So there's things at each of those stages every year. And it's kind of a big machine that just turns continuously.
A lot of questions. Go ahead.
So, a lot of young people are trained in horticulture, still can grow berries, and flax, What 3 kinds of classes would an undergraduate or master's student have today for you to be able to use them in this transition between horticulture and molecular methods? What real 3 things would you look for a student would have to kind of do a little bit of both and work for in that environment?
3 classes. It depends where they're going to be working. If
on that marriage between screening, pathology, horticulture, and moving into some molecular methods.
I think you would need at least a basic genetics class, and I think that would probably be enough to sort of at least equip you to deal with plant breeding and with molecular biology. I think a plant propagation class, something like that, could be really helpful. And it depends kind of what role they're going to play. I think plant nutrition could be important for the growing aspect of it. I also, pathology has been a big part of my career, and it's such an important trait in a lot of programs that I think a plant pathology class would be pretty useful. I guess that's 4, but. What else?
So you do GWAS for all traits or some specific traits that need to be?
So the majority of traits we do GWAS. So we actually have a panel of about 200 selections that we've sequenced entirely. And we have any trait that varies in that group, we phenotype and run GWAS on. There are some traits that are very rare. If only 1 or 2 things in the population have it, then we can't use that approach. And then we'll use a segregating population instead.
So when you do GWAS, do you use what in specific software to analyze your GWAS?
I give it to our molecular biology manager, and he gives me that graph. Anyone else?
So
What are the incentives for a Driscoll grower to put out that one of those plots for you the year before it's commercialized? Are you paying them, or are they getting premiums?
So that 2% trial,
Yeah.
so that is in their contracts. They're required to do that if we ask them. So it depends, the level of enthusiasm for that depends a lot on the location and the variety you're trying to put out. Generally in Watsonville, I have more people asking for it than I have plants to give out. In some places like Baja, for whatever reason, I think partly because they've been really profitable with the varieties they have, they're just like, I'm fine, leave me alone, don't mess with what I'm doing. And we send them plants and then they conveniently forget about them, and then they're like, oh yeah, those things molding in the cooler, we should have planted those, shouldn't we? So I have to twist some arms sometimes in some places, but generally there's interest in new varieties. And I try to mix it up a little bit if it's something that's like, people are really interested in, try to mix it up a little bit on who gets what. We could do a straight 2%, but there's some really small growers. So, if you have 30 acres and you're going to do 2% of that, you don't learn very much by farming a couple rows, So I usually give those guys the option, but I don't try to make anybody do it on that scale.
Other questions? I got a bunch, but I want to.
Sample for perennial.
Yeah, we have. It's part of the GCMS.
Come on, let us know what that is. It's a very sexy molecule.
Why do you think so?
Because I work with tomatoes, and we have people blind tasting the high perennial varieties. Students are literally trying to put them in their pockets so I
So furaniol and mesofuran, which are two related compounds, are two of the most dominant volatiles in strawberry. And they kind of have a, I don't know, like a burnt sugar caramel kind of aroma on their own. You don't really get that from a strawberry. But if you have a variety that's low in furaniol and mesofuran, it doesn't taste like a strawberry anymore. I mean, you get really weird tasting things, and most people don't like them. We actually have varieties that make no mesofuran, and they tend to have lower furaniol as well. And we've found that when you do flavor tests, almost nobody prefers the things without mesofuran. But the people who do feel very strongly about it, and almost without exception, you ask them why they liked those ones, and they go, well, I don't like strawberries, but those ones were okay. So, it's really inherent to strawberries. And, one of the things we've done with markers is we've used markers to avoid selecting things that don't make mesofuran, because most people don't like them. Furaniol, the control's a little more quantitative. It's harder to do with a single marker, but it's really important. Anybody else?
If something makes it to stage 4 in the selection process and then isn't resistant to a certain pathogen, do you ever revisit any of those cultivars that just didn't make it all the way?
Sometimes. The general philosophy of the breeding program, I always say to the breeders I supervise, is just never look back, because there's just so much in the pipeline that you can't afford to dwell on losers, So usually I just say, if it doesn't make it, it doesn't make it. We got better stuff coming. But there are cases where, if something has been dropped for a very narrow reason, I sometimes will make an extra effort to kind of shop it around to other geographic locations, or talk to people and say, we really liked this, but we had problems with Colletotrichum gliosporioides, but we know you don't have that, so maybe it would work there. So there are a few kind of near misses that we have gone a little further. The other thing is the cross-trialing stage, where, at stage 1, where we're seeing stuff from different breeding programs, sometimes something will wind up advancing in 2 breeding programs. So, for example, we have a variety we were talking about today, Fortaleza, came out of the Watsonville breeding program, got dropped in Watsonville, but actually advanced in the Santa Maria breeding program. And so we had this situation where a Watsonville variety was being commercialized in Santa Maria. And at that point I said, you know, this thing looks impressive enough in Santa Maria, why don't we look at it again in Watsonville? And in the end, we decided to go ahead and commercialize it in Watsonville. A lot of the same concerns that resulted in being dropped in the first place still existed. But I think, you know, seeing it at a large scale in Santa Maria, we felt like, you know, the problems are real, but we're dealing with them there. We can probably deal with them in Watsonville too. So there's a lot of kind of close judgment calls, and you sort of have to, I don't know, I always imagined myself as a breeder that I would have a really tough time dropping things, but I actually, most of the time it feels really good. It's like, you know, it's, because it's part of the process. It's like, you know, you're like being a breeder. But once in a while, there's a few things that you've kind of, you know, gotten attached to, and it's really hard to let them go, even though in your heart, you know, that, you know, they really don't have a future. But there's been a few really good tasting things that were really sick plants, and it's like, well, let's look at it one more year because I want to eat it. So.
Peter?
So with all these cultivars or varieties that you have, that you're breeding and releasing, they don't put names on the boxes. So how do people know, or how do you know?
Most people don't. The one exception to that is in Europe, in the UK, it's almost mandatory in the UK, and in the Netherlands and Belgium, they frequently will put it on the package. A lot of retailers demand that. Here we've avoided it, and it's really because we turn over varieties so fast that we don't want to get anybody getting attached to things, because it makes it hard to replace it. I was talking to these guys earlier. You know, the hood strawberry in the Pacific Northwest, or the Marionberry in the Pacific Northwest, they've now they built a brand around those varieties, and now they're in a situation where they have better varieties and they can't replace them because people go looking specifically for those cultivars. And so what we hope is that, you know, the Driscoll's brand is what people look for and recognize, and it's our job to make sure that we associate only the best cultivars with that. You know, there are things, you know, we for a long time we've had two labels. We've had the yellow conventional label and the green organic label, and that's it. We are playing around with concepts of a premium label, because there are some varieties which, you know, are not going to make it if you just stick it in a regular label. You get the regular price for it. You know, you've got low yielding, super sweet varieties. I've got one of those in Watsonville this year, and if everything goes right, you may actually be able to find it in supermarkets. Actually, it may be there now, but it's not under a special label. But there's 36 acres of it, 38 acres, something like that. So. And then we have the pink strawberry, which you'd know if you saw. So.
You might have answered my question, but you know, you're talking about how quickly we go through a variety. You said 3.3 years is the average life of a Driscoll's variety. So for the students, there's a variety in Europe that they grow in protected culture, so greenhouses, tunnels. It's called El Santa, and it's been popular for over 30 years.
The cross was made the same year I was born, 1974. So.
Yeah. So how do you explain what we're doing here with 3.3 years per variety and what's going on with El Santa?
I think there's a couple things going on, and some of it is just sort of a difference in philosophy. So like our philosophy is, you know, we have just a couple growing systems, and we solve our problems with different varieties. Their philosophy is we have just a few varieties and we solve our problems with growing systems. And so they have, you know, they might have, you know, well, they've got all these different plant types. They've got misted tips, they've got waiting bed, they've got, you know, cold store tray plants, you know, all these things, and they plant them at different times and they manipulate them to get production when they want. And they've done all these things with the same variety. And I think that in some ways, I mean, they would make a different argument probably, but I feel like they sort of painted themselves into a corner because they developed all these systems around Alsanta. So as soon as you try to put something else in it, nothing's going to work as well as Alsanta because Alsanta, the whole system was designed for Alsanta. And so it's sort of a self-perpetuating thing. It's like you try to replace Alsanta and you go, oh, well, you know, it works as a 68 plant, but it doesn't work as a waiting bed, so I'm just going to stick with Alsanta. And while that's happening, the consumer, particularly in the Netherlands and to some extent in other Northern European countries, has gotten really used to Alsanta, which is a little kind of lumpy orange berry. It tastes okay, but not great. But that's like, you know, I mean, they've been growing it since the 70s, so it's like most of the people, that's what they've spent their entire life eating. And so that's what a strawberry is. And so it makes it really hard to introduce something that is even slightly different, because people are like, well, something's wrong with this strawberry. It's slowly happening, I think, because now other varieties have gotten so far but beyond Alsanta in a lot of regards. But it's been really, really slow. I remember one of my first trips to Europe, we were trying to recruit growers in the Netherlands, and we had a guy who grew a trial of a variety, a Driscoll variety called a Mesty, and I went out and looked at it, and I was like, well, how's it doing? And he said, well, you know, it's yields more than Alsanta, the fruit's bigger than Alsanta, the flavor's better than Alsanta, it ships better than Alsanta, it's easier to pick than Alsanta. I said, you can grow more next year? He said, no, the stores all want Alsanta. So, which as a breeder is a little disheartening, because it's kind of like, why bother? But
It's quarter after. I'm sorry to keep you guys here this long, but if you want to stay and have further questions for Dr. Stewart, please do. Otherwise, please hand in your evaluations, and let's thank again Dr. Stewart for an outstanding seminar.
thanks for having me.